extremely interesting. I've learned a ton. I really like this idea of an AI and ag crossover. I'm going to be changing tacks a little bit with respect to the previous talks, primarily because I'm coming at the problem more from the statistics, I guess more broadly, artificial intelligence side of things. And I'm realizing that if I introduced more robotics and sensors into my work, I'd have a lot more cool images and videos to share. But what I'll be talking about today is a bit of model development that we've been doing to try to improve the statistical models that are typically used for genomic prediction, to be able to better account for information that we've collected in various sources elsewhere. So I guess it's an additional change of tacks compared to the previous talks, is that I'll be talking here about genomic data primarily. And so the work I'll present today is primarily the work that was done by a former postdoc of mine, Fanny Molandin, who was co-supervised with our colleague Pascal Croiseau. So to start, just a quick word about INRAE, which is the research institute in France where I work. So this is the French National Research Institute for Agriculture, Environment, and Food. There's several different research centers scattered throughout France. I work at one in a city called Jouy-en-Josas, which is just outside of the Paris area. It's a fairly large research center that has over 1,500 different staff members and has a fairly wide spectrum of research activities that are covered, going from animal biology to microbiology, data science, and more widely systems biology. So within the INRAE Research Center at Jouy, I'm part of the Animal Genetics and Integrative Biology unit, which focuses on various themes that are related to animal genetics, notably trying to understand and characterize genetic variability in animal populations, as well as the construction of phenotypes that are of agricultural relevance and how they might interact with various environments. And of course, as with everybody here, I'm thinking about how these animal genetics tools can go towards contributing to the agroecological transition. So with that sort of general introduction in place, I'll move on to the sort of specific objective of what we're looking at doing in terms of genomic selection. And this might be a topic that is familiar to many of you in the audience, but generally speaking, what we're seeking to do is here in terms of animals, but a similar logic applies to plants as well, is to be able to select the best possible animals to use for the reproduction of future generations with an aim to obtaining genetic improvement of the population for whatever the trait of interest might be. And I could cover a fairly large spectrum of possibilities. So the idea here is that genomic selection is going to be making use of genetic variability to try to understand how that variability and to what extent it contributes to the variability of our traits of interest. So for this purpose, the most commonly used type of genetic variability is something called a single nucleotide polymorphism, a SNP, which represents a single point mutation in the genome that is sort of represented by the schematic illustration here on the slide. And one of the reasons that these SNPs are particularly widely used for genomic selection is because we can now measure them on a fairly large scale fairly easily. And so the idea here is that with advances in technologies over recent years, we've been able to go from using low to medium to high density genotyping chips to allow us to get information on what the specific allele is for an observed SNP in a particular individual, which gave us on the order of tens to hundreds of thousands of SNPs for each individual. And then more recently, as costs have continued to decrease, we've been able to go on to the use of whole genome sequencing. So here we're looking at many tens of millions of SNPs that would be able to be characterized for a single individual. So what happens once we have access to these data? Well, as with other prediction models that we've seen in the previous speakers' talks, there'll be a phase where there's a model that has to be trained on an observed set of data for which we have observed traits of interest. So for example, feed efficiency in pigs. So this model will be trained on the reference population, and once the model itself is in place, then it can be deployed on a new set of individuals for which only genotypes are available in this prediction phase. And so that would be performed on a validation population to obtain those predicted values of the trait that we're interested in. So if we say it differently, the same general idea in terms of the statistics, what we're doing is we're given a training set of data, and this training set is made up of our fully labeled data where we have our observations of our trait of interest. We have this large vector of tens of thousands up to millions of genotypes that have been characterized at the genome-wide level. And then typically there's a set of covariates that must be accounted for in the model as well. So age, location, sex, just to name a couple of examples. And of course, as I mentioned previously, we'll be looking to predict an unobserved trait in our future individual that has the corresponding values of X and Z that go along with it. So genomic selection is probably familiar to many or most of you, as I said, because it was introduced several years ago now, early 2000s, and has actually been fairly successfully implemented in many plant and animal breeds for a variety of traits. Many that are focused to production related traits, but also with respect to health or with respect to robustness or adaptation to climate change related traits. So one reason that these models have been so successful is because we've been able to see at least modest gains in predictions that can yield fairly large economic impacts in practice. And that's because we can reduce the generation interval, we reduce cost and labor for phenotyping, and this partly explains why genomic selection models have been fairly widely deployed today. Okay, so in terms of someone coming at this problem from a statistics perspective, there are obviously a lot of challenges that come with analyzing genomic level data for these prediction models. And I'm mentioning just a couple here on this slide as a sort of teaser of the types of things that cause issues for a statistician that's working to develop these models. One aspect that can sometimes cause a bit of a headache, let's say, is that there's non-random association between alleles, between our SNPs that are close together within the genome. And this is a phenomenon that's referred to as linkage disequilibrium, which means for a statistician that there are many variables in my genotyping data set that are very highly correlated with one another. And that can cause issues in terms of the statistical modeling. A second challenge is that many of the complex traits we're primarily interested in are highly polygenic. So there's not necessarily a single gene or single SNP that will be responsible for the majority of the variability of that trait. It would be scattered in terms of little effects that are scattered all about the genome. And so that means that it's not a matter of just finding the one SNP that's of interest. It's sort of, we have to account for this fact that there are many small effects that might contribute to the construction of a particular trait. And finally, it maybe goes without saying that we're working in a difficult setting in terms of the statistics because we have many, many more variables. So here are the SNPs. Then we do animals that we have genotypes for, or our observations. And this is the classic problem of the curse of dimensionality that we hear about, where our variables are much more highly numerous than our individuals. So why is that a problem? As with many of the other data that we've seen in the previous talks, if you fit a model that has way more predictors than observations, well, you run into problems related to overfitting with respect to poor generalizability of your model. So being able to reapply it to a future data set will work much less well. And there are additionally problems with the estimation of the model from a statistical point of view. So that's the one extreme. On the other hand, if we try to come back to having a sort of small pre-identified subset of variables or SNPs to focus only on those for the purposes of constructing a prediction model, typically what we see in practice is that leads to fairly poor predictions. So as one example, a sort of straightforward approach might be to focus only on genotypes that one by one have been identified as being highly associated with our trait of interest, so in a GWAS type study. But that's sort of the other extreme where we've removed too many of those small effect genetic variants that actually do contribute something to the architecture of the complex trait we're interested in. So all that means that we're looking at trying to balance the sort of computational and statistical feasibility of our model with something that's sufficiently close to what we can call a biologically realistic assumption. So we need to find a balance, strike a balance between these two aspects. And that brings me to the major driving question of the work that I'm going to present to you today, which is this idea that we'd like to understand whether genomic prediction models could potentially be improved if we were to better account for the things we already know about the genome, and in particular, the function of certain regions of the genome. And I'll get into a little more detail about what this prior knowledge could represent, but the idea is that we have today access to tons of data that are available in public databases that have come from the many scientific studies that have been undertaken in recent years on the function of the genome in livestock species. And the idea here is we'd like to be able to better account for this prior knowledge when constructing our new genomic prediction models. So to give a little context to why specifically we were interested in looking at this use of prior biological knowledge in genomic prediction models, I'll talk a little bit about a European project that I'm involved in that's called Geneswitch, which is an acronym for the Regulatory Genome of Swine and Chicken, Functional Annotation During Development. This is a project that's co-led by my colleagues Elisabeth de Joffre and Hervé Acloc here in France. And as part of this Geneswitch project, one of the major outputs will be the construction of a large number of richly annotated functional maps of both the pig and chicken genomes. And what they did within this project is actually to generate a large number of molecular assays at different levels. So at different levels of development, starting in early late organogenesis to newborn or hatched pigs and chickens or adults. We also looked at different sexes, different tissues, and then, as I said, different layers of molecular assays. So transcriptome with RNA-seq data, chromatin accessibility with ATAC-seq data, histone marks with ChIP-seq data, small RNA-seq data, methylation, and the three-dimensional chromatin maps with Hi-C data. That means we have all these different layers that can kind of build onto the scaffolding of the genome that we're familiar with and can inform us about which particular regions of the genome may be most pertinent to upweight in some sense in a genomic prediction model. So we're seeking to be able to integrate this functional information on the one hand with phenotypic and genotypic data on the other to be able to ideally achieve greater power in our genomic prediction models, as well as to potentially improve their interpretability. So to upweight genetic variants that are more likely to actually play a functional role in the construction of those traits. So all that was a great idea, but the major question then was, how will we go about integrating this type of information into the model? So to explain our approach to the answer to that particular question, we have to get back to basics and just think about what the fact that most genomic prediction models are actually constructed on a basic multiple linear regression model, which looks something like that shown here on the slide. So as we already saw, we have a vector of traits Y that we're looking to predict. We may be accounting for some set of covariates. And the primary quantity that I'll be interested in looking at today is this X beta, where we're looking at the genotypes multiplied by this vector of genetic effect parameters. And what we'll be doing is looking at how we can play around with our assumptions of what this vector beta looks like, or genetic effect parameters, to be able to account for this prior functional knowledge. So if you hear me saying this word prior knowledge, those of you who have heard a little bit about Bayesian methods might have perked up a bit and recognize the terminology. And in fact, Bayesian methods were a set that we were particularly interested in here because it so naturally allows for the incorporation of prior knowledge in the form of prior distributions with the likelihood or the evidence that's provided by the data we have. So very broadly speaking, what Bayesian methods are looking to do is to balance between these prior expectations with the evidence that's provided to be able to have a posterior distribution of the parameters that we're looking to understand in the model that allow us to have this sort of balance between the two. So if we're thinking about Bayesian methods for genomic prediction, then that means that what we'll be looking at is this likelihood on the one hand and our prior distributions on the other. And just for reference, on the left here for the likelihood, we see a fairly standard likelihood. Here would be a Gaussian distribution if we've assumed that the errors of our model are normally distributed. And what we see here in red is that the genetic effect parameters that I'm going to be primarily interested in, the beta J's, are represented here in pink. And we'll also have a set of prior distributions here on the right that will describe how I expect those parameters to vary themselves. And we see once again my genetic effect parameters that appear here on the right. So what I'll be wanting to play around with a little bit is how exactly should we define this prior distribution on the right for my vector beta J, so as to be able to encode realistic biological assumptions about how I expect my genetic variants to contribute to the trait, while at the same time being able to permit a computationally feasible solution to estimate this model. So which prior to use for this vector beta J has been a focus of many papers linked to model development in this space. And generally we'll sort of get at trying to understand to what extent we want to force some of the genetic variants in our model to have exactly 0 coefficients. So in effect, performing a bit of variable selection so as to be able to have a more parsimonious model, versus trying to allow each genetic variant to have its own particular effects, its own unique effect within the model. And this is just a quick visualization of a couple of different options that have been proposed as a prior distribution for these beta J's. And in fact, if you look through the various models that have found great success in the literature that have been proposed in the meantime, what we see is sort of a pattern. So the major model that's used today for genomic prediction models is the GBLUP because of its computational efficiency. It tends to work well. It's a simple model that has shown good performance in practice, and so has often been deployed in routine. And this GBLUP model assumes that my vector of genetic parameters beta I here follow all the same normal distribution. So here a Gaussian distribution that has a particular variance sigma squared beta that's shared by all of the genetic variants within my data set. So it's a fairly strong assumption in terms of the reality, the biological reality of how traits are constructed, but it tends to work quite well in practice. And what happened is that in the years since, there's been an ever-widening development of a set of models called the Bayesian alphabet, in which various authors have proposed playing around with how we can model this prior distribution of beta so as to be able to provide a more realistic solution. So the first two of the Bayesian alphabet that were introduced were the Bayes A and Bayes B, which you'll notice differ primarily from the GBLUP model in the fact that the variances of my genetic effects beta now have their own subscript here. So I have a beta I here in my sigma squared, my variances of these normal distributions, which means that each genetic variance is now allowed to have its own variance. Right, so it's a more flexible model than the GBLUP model, but it's also quite a bit more computationally intensive because that means I've now multiplied the number of parameters in my model considerably. So to address that sort of computational difficulty, there were further extensions. We can name the Bayes C and Bayes C prime models, which have tried to get at the problem a little bit differently. They've had a beta in which we have a big portion here that is essentially set to 0. So a fairly significant number of genetic variants will have absolutely no contribution to my model, whereas the others will follow a sort of basic normal distribution and so on. And so these play around with how, to what extent we'll be able to set the percentage of genetic variants to have a null effect or not in the model. Okay, so those are all good and well. About 10 years ago, there was a further extension to this Bayesian alphabet family of models. It was called Bayesar. And Bayesar essentially tried to cut it both ways, let's say. They wanted a little bit of flexibility, but not too much in the model. And what they did is assume that these genetic variants now follow a mixture of 4 different types of effects. And those effects are null, small, medium, or large. So these variances are essentially fixed in the model. And what we're going to do is allow some flexibility now that we have these null, small, medium, large effects, but not so much flexibility that the model is difficult to estimate. So this essentially gets at trying to model my genetic variants in terms of a proportion of null, low, medium, and high effects. And it turns out that this Bayesar model actually worked quite well in practice. It's more computationally intensive than a standard GBLUP approach, but works well enough that it's a solid competitor in a lot of situations in practice. So the Bayesar is a little bit of a reference model for us in terms of where we're going to go here. One reason for that is that the Bayesar model was itself extended to be able to account for a notion of some prior knowledge based on functional annotations by MacLeod et al. In 2016. So now we've again further expanded the Bayesian alphabet to have a Bayesar C model here, the C being a categorization of SNPs in the model. And what MacLeod and colleagues proposed is essentially to break the genome up into the individual Bayesar models. So the idea being that each genetic variant, each SNP, can be assigned to a single category of annotations. So in my illustration here, I have 3 particular annotations that are available, and a SNP is assigned to one and only one of these annotations. And because these annotations are completely disjoint from one another, that means that within each annotation, I can fit an independent Bayesar model to have a null, small, medium, or large effect for each genetic variant. And that means that I'm essentially looking at varying the proportion of large, medium, small, null effects within each of these different categories of annotation. So that gets us to the Bayesar C and to being able to account for these prior categories of annotation. And these prior categories of annotation are sort of defined voluntarily in a very broad way. So we could call an annotation a set of genetic variants that are neighboring genes that are known to play a role in lactation, for example. Or another annotation could be SNPs that have been flagged in previous GWAS studies as another example. So that all works well and fine once we have a fairly small number of annotations and once there's no overlap among those annotations. But as our biological knowledge becomes more complex, the story isn't quite so clear anymore, as we might start to have more and more overlaps between potentially interesting prior categories of annotation. Which is where we get at when we want to define a possible way for accounting for those increasingly complex annotations. So going from Bayesar to Bayesar C and beyond. So if we come back just a second to the Bayesar approach that they've already mentioned, if we look at things in terms of a simple illustration of the idea here, the Bayesar approach, as I said a moment ago, is focused only on using the genotype data itself to predict whatever our trait of interest is. And the Bayesar model gets around this balance between computational efficiency and biologically realistic assumptions in essentially modeling a proportion of genetic variants that correspond to each of these 4 levels of effect within the model. We saw just a second ago the Bayes-RC approach is going to take the approach that we have these different annotation categories that are available. Here, an orange category that might correspond to SNPs that were GWAS hits. A yellow category is variants that are known to exist in a particular database of interest. And we'll be modeling those proportions of high, medium, low, and null SNPs independently within each annotation. Now, if we get at this idea that we have our multiple annotations that might arise from our multi-level, multi-tissue assays coming from, for example, the Geneswitch project, we'll see that very quickly we'll arrive at situations where we'll have SNPs that have multiple annotations. And that immediately runs into a problem for the Bayes-RC approach, which necessarily has to factorize our model into these independent categories. So this was the primary issue that we wanted to account for with a novel set of models that we developed. And the 2 set of models that we developed are part of a package that, again, further extends this Bayesian alphabet family. So Bayes-RC-O for overlapping annotations. But in fact, the Bayes-RC-O software we developed includes 2 distinct models because in our minds, there were 2 distinct hypotheses or assumptions that needed to be accounted for. The first is that when there are multi-annotated SNPs, depending on what type of annotation is included in the model to be able to provide prior information about genetic variants, it may be that overlapping annotations reflect in some sense a bit of ambiguity or uncertainty, whereas the highlighted example here on the slide may be that we don't really know whether the SNP in question should belong to or is more informative in the accessible chromatin annotation or in the animal QTL-DB annotation. Maybe that we've put both here in the model, but we're not really sure whether one or the other makes more sense. And this will be the issue that'll be approached by the Bayes-RC Pi model. On the other hand, depending again on the annotations we've chosen to use in the model, it could be that the more a SNP has annotations, the more likely it is that I think it's an informative SNP, and I sort of want to drive a greater weight being assigned to such variants. So a sort of cumulative approach to combining multiple annotations for those multi-annotated SNPs. And that's the approach that will be taken by the Bayes-RC Plus model. So that means we have 2 different hypotheses for 2 different models that aren't necessarily comparable because it really reflects whether we consider our multiple annotations to reflect an added confidence, as I said, for this Bayes-RC Plus approach versus uncertainty that we want to try to resolve within the model itself, which would be the Bayes-RC Pi approach. So for both cases, we're explicitly handling the fact that we have potentially overlapping annotations for a SNP. If you think about an annotation matrix being something like a binary 0-1 matrix, it's essentially, do we have more than one for each SNP or not? And we'll get at that using the equations that I've presented here on the slide, but I won't comment in detail. Okay, so yeah, whenever we're developing models, new models in practice, whether it's a Bayesian model, a machine learning model, or otherwise, the initial step to take to make sure our model is doing what we want it to do is typically to simulate some data, try to come up with a situation in which we control fairly well the data, what the true QTLs are, that contribute to our simulated phenotype so that we can determine to what extent our model is providing good predictions on the one hand and is accurately upweighting those truly causal variants that we've actually simulated in the data. There are loads of ways to think about how to simulate data in such a setting, bearing in mind that in any case, we're always operating in this overly simplistic situation, right? That only partially reflects what's really going on in real data. So to try to keep things, let's say, as close to realistic as possible in some sense, what we've done here in our simulations to validate the developed approaches is to start off with a set of real data. So we had some real genotypes from a low density chip in cattle, for which we had data available on 2,500 animals. And what we did then is we essentially created a situation in which we were able to randomly select among the genotypes a set of true large QTLs, so genetic variants with large effects that were defined using a percentage of the total additive variance that was fixed in the simulations, as well as a set of sort of medium QTLs and low effect SNPs to try to spread out the effects within the genome and try to mimic the sort of polygenic architecture that's typical of most complex traits. We've played around with different levels of heritability to make the situation more or less difficult. And then because results always depend on which specific data set you generated, because there's some randomness that's associated with each simulation, we then repeat this whole process 50 times to be able to understand and characterize the variability that we see in results across simulations. So one thing that's additionally a little bit tricky in this scenario is not only are we simulating phenotypes based on a given set of genotypes, but we also wanted to play around with trying to understand to what extent the informativeness of annotations was important. So in other words, if we use annotations, so overlapping annotations for our SNPs that we've used in our simulations, if we use annotations that are totally off the mark, does that penalize us, or is the model able to bounce back even so? Because we're in a Bayesian framework where these annotations are part of the prior knowledge, the question was whether the evidence provided by the likelihood would be able to successfully avoid being brought down by poor annotations. And on the other extreme, if we have strongly informative annotations, so those that are strongly enriched where we have a lot of our known QTLs that are flagged as being annotated in a particular category, how much better does it help us do in terms of our modeling? And so that's what we've done here with our scenarios A, B, and C that we've simulated. We've generated these annotation categories to be able to evaluate this point. But I won't spend too much time on the simulations, but I do want to highlight just maybe 2 or 3 interesting results that we saw that sort of confirm that the model was doing more or less what we expect it to do. So this first result that I'm showing is the results of looking at the difference in terms of the accuracy of the model in our validation population. So as always, we have a training population that was separate from the validation population. And in each case, I'm looking at the difference in these novel models that account for annotations versus a baseline Bayesar model. So versus a model in which no annotations at all are used. And in these graphics I have on the left, a sort of low heritability situation, a heritability of 20%, and on the right, high heritability, but the trends are similar. So we see the 3 annotation scenarios, A, B, and C on the x-axis. A being a situation where annotations are supposed to be highly informative, and C a much more mixed case. And what we see here is that the Bayes-RC and Bayes-RC-O approaches that we've developed here tend to perform better than the Bayes-R approach, so have positive values in these highly informative settings, so scenario A. In scenario B, which is a bit of a mixed case, we have a better performance with higher heritability, but that seems less marked for the lower heritability case. And then we do see that there are cases where having overly fuzzy, let's say, annotations that mix in non-informative annotations with the rest can actually lead to a detrimental effect on the performance of the model. So we do worse in these cases when we use the Bayes-RC and Bayes-RC-O methods than we did if we had just used a standard Bayes-R approach. So that suggests that there are cases where we should be careful about what annotations are included in a model. And it's not a free-for-all to be able to put in everything that we have available to us. Briefly, I also wanted to double check two things. The first is that for the model that handles uncertainty, this Bayes-RC pi model, is it accurately able to assign an informative annotations to QTLs when they have multiple annotations? The answer is yes, because we can see in the plot here on the left that when there are truly large QTL effects, we see that they tend to have higher probabilities of being assigned to an informative annotation. And it's a similar image that we see on the right as well. This Bayes-RC pi model is going to play a game of trying to assign multiply annotated SNPs to the most informative annotation. And we see that that does appear to be functioning in the case of the Bayes-RC pi approach. With respect to Bayes-RC plus approach, when we're checking if it's doing what we expect it to do, the idea is that as the number of annotations for a given SNP increases, we expect the model to be increasing the weight that's attributed to that SNP in the model, or in other words, increasing its log posterior variance. And here, whether we focus on the large QTLs on the top or the medium QTLs on the bottom, we're seeing indeed the model is trending towards boosting genetic variants that have larger numbers of annotations associated with them. So in some sense, the Bayes-RC plus model is also doing what is expected of it. So I wanted to also do a quick illustration of how these models work on a set of real data. This is a sort of initial validation of the models on an independent set of data that we did towards the end of this portion of the project. So what we did is we looked at data that were available in a back cross population of growing pigs. These were data from my colleague Hélène Gilbert at INRAE in Toulouse in France, she was working with a collection of about 1,300 back cross pigs. They were 3/4 large white and 1/4 Creole. That were genetically related sows that were sired with a set of 10 different boars. So these were data in which there were 60K SNP genotypes that were performed using an Illumina porcine bead chip array. And what's interesting is that, again, I think Claire said something about how these livestock applications often allow for interesting questions in terms of cross-validation structuring that are quite unique to the ag sector. And this was a case where, because the sows were from 10 different families, 10 different boars, we put into place a 10-fold cross-validation procedure using the sibling structuring that was available to us. So here the traits that we'll be looking at were pre-corrected for a couple different covariates that are not of primary interest here today. And I'll focus in particular on two different traits that we looked at in more detail. The first being average daily weight gain, or ADG, and the second being back fat thickness at 23 weeks, or BFT. Okay, so we have our genotypes, we have our traits in place, and then the next question was, what shall we use for annotations to try to bump up the information content of certain genetic variants in our data set? Actually, one question you might ask yourself is why here I'm not rediscussing the Geneswitch molecular assays that I presented at the start of the talk. And here is a purely opportunistic reason. The work I'm presenting here was actually conducted prior to the availability of those molecular assays in Geneswitch. So now that they are becoming available and coming out bit by bit as we speak, we'll be able to make use of them to evaluate the differences in these results. But for the time being, what we used is annotations that were constructed from a public database, so PigQTLDB, which essentially is a catalog of GWAS results for a variety of different traits and trait sub-hierarchies. And so here what we looked at is GWAS results for a set of 11 different trait subhierarchies that are listed here on the slide. So we, if a genetic variant was identified for anatomy related traits in GWAS, then it belongs to the anatomy category and so on. And to account for this idea of having linkage disequilibrium, so highly correlated genotypes at neighboring loci, nearest up and downstream neighboring markers were also associated with these annotation categories. So I should have done a drum roll or something, but the results of these different methods on the ADG and BFT results are shown in this table. And what we have is the results for the Bayesar approach first in the leftmost column. And we see here that using this 10-fold sibling structured cross-validation, the correlation for the validation population was 21% for ADG and 26% for BFT. So these are traits that are have fairly low heritability, somewhat difficult to predict, and BFT in particular had quite large variability among the different folds of the cross-validation procedure. And then, of course, what we're most interested in looking at is how do we improve, or on the opposite, how do we worsen our performances when we make use of these PigQTLDB annotations. And what's quite interesting is we see that for the ADG trait, we tend to have fairly modest improvements, regardless of which overlapping annotation model is used. So between about 1.5 to 1.7 correlation points of improvement. Whereas for the BFT trait, we actually saw a decrease in performance. So the Bayesar CPI model in particular lost a full correlation point with respect to the Bayesar model. So that's sort of a mixed bag here. We have a modest improvement for one trait and a modest worsening for the other. That said, what's interesting is that if we take the specific example of the Bayesar CPI approach, this is a model that allows us to dig into which of the particular annotation categories appears to be the most informative, so contains the most or the highest proportion of large and medium effect SNPs. What's quite interesting is we see that for this ADG trait, the behavioral, confirmation, and litter traits are sort of at the top ranking in terms of proportion of high and medium traits, which is somewhat interesting that to see at the start. And we see also that the ordering of annotation categories is quite different between our two traits. So ADG, where arguably there was a slight improvement that was observed on the one hand, versus BFT, where we actually saw a worse performance. So some conclusions to wrap up about this, this work that focused on incorporating annotations with the Bayesar CO model. This Bayesar CO software includes these two novel models, Bayesar CPI and Bayesar C+. Bayesar CPI seeks to assign informative annotations to these SNPs for which we have lots of prior knowledge to be able to better account for the uncertainty we might have. Whereas in situations where all this prior knowledge leads to an accumulation of confidence that the variant in question should be included in the model, we have the Bayesar C+ approach that does successfully upweight such markers. So all that, I guess it's a little bit of a mixed bag in terms of conclusions, because what we saw, whether in simulations or in real data, is that in reality, we tended to have fairly modest improvements in prediction on the order of 1 to 2 points of correlation when we incorporated biological annotations. So there's an idea that there were very slight improvements in predictions. We did see improvements in rankings of QTLs, particularly in simulations. And particularly so when the annotations were highly informative. So that does suggest that there could be some room there for interest in digging further into how annotations can be incorporated in such models. Again, it's a very slight improvement only in some cases for real data. And the other point I should mention that I didn't delve too much into here is the fact that not only is the choice of source of annotation important, but even strategies for how to construct categories appear to impact results. So there's a lot of work there, I think, to think about how we can best make use of all this mass of prior knowledge that exists and is sort of waiting to be exploited. So just one quick slide of take-home message, and then I'll wrap it up. I wanted to come back to the original question I asked at the start of my talk, which was, can genomic prediction models be improved by accounting for this biological knowledge about the function of certain regions of the genome? So I think what I've shown you today is the answer is yes, but yes, sometimes. Most certainly can't unambiguously say that in all cases such a strategy would be beneficial. And I think there are several different levers of action to perhaps come up with a more concrete answer to that question. There are obviously different ways of considering the modeling itself. So developing other models, more sophisticated models, delving more into machine learning approaches that can incorporate annotations and so on. In my mind, this isn't necessarily the major blocking point. I think that the Bayesar CO approach we have here to account for overlapping annotation categories is a promising modeling setup, and it actually sets the stage quite nicely for extensions to handle quantitative annotations rather than categorical. The other point is that a lot of the simulations and real data analyses I've discussed today focused on mid-density genotyping chips. And it's in fact probably the case that capitalizing on annotation maps like those generated by Geneswitch will require whole genome sequencing level resolution. Another point we've been thinking about a bit in our group is the validation data. So in fact, the idea that it's possible that greater potential gains may be seen when genomic prediction is performed on more genetically distant populations, for example, crossbreed as one possibility. So that's something that we want to look into and try to evaluate. A fourth point is that of the traits themselves. So obviously the potential gain for traits that are already well predicted by existing models is likely less. So the different heritability of traits, the underlying genetic architecture are two aspects that are quite important to consider, as well as the link with a given trait and a set of annotations. How tightly connected are they? Which brings me to the last point, which is one I've already alluded to a bit in my talk, which is what annotations are most pertinent to use in this scenario. So we have, again, access to many, many different data sets that are linked to different types of molecular assays, but which assays are the most pertinent for this task and in which tissues? So these are open questions that we're continuing to work on in our group, and I think have been the focus of several different developments in recent years. So we'll have to continue those efforts to try to better answer the question of incorporating this knowledge into genomic prediction models. So with that, thank you for your attention, and the reference for the methods paper that's associated with this work, as well as the open source code that Fanny put in place for the Bayesar CO model that's located at the FANG GitHub. Thank you.
Thank you very much, Andrea, for your great talk. It was very pedagogical. So Much appreciated. Thank you very much. We have a first question in the chat, so I will call out for you. Did you look at how well these approaches worked when you randomize the phenotype? So, for example, the random outcomes.
Oh, no, you mean like just to see whether it changes things on a totally random phenotype?
I think so. I did not ask the question
no,
myself,
I didn't. Yeah, I mean, I wonder whether the idea would be to see whether if you have totally random phenotypes and you inject some annotations that then would be completely non-meaningful, whether that would change the predictions or not, or a selection bias issue. I see. No, I did not look at that. That would be interesting to look at.
Okay, thank you. I have another question. How did the GBLUP model perform in the real data? Did you try this?
It was, it was tried, and I don't have the results in mind. So I can't give you the specifics on the comparison between Bayesar, for example, and GBLUP, but it was done.
Thank you very much. So yeah, we have another question. How does the Bayes RCOs compare in terms of computation compared to the Bayesar model?
Yeah, that's a good question. Because it's true already as it is, Bayesar, it's a fairly good balance between this biological realism and computational feasibility, but the computational time is much longer than a classic GBLUP. And when we go into trying to include overlapping annotations in these Bayes RCO approaches, there is some sort of double sums that are happening. So in fact, the Bayes RC plus model, which tries to account for this cumulative contribution of multiple annotations, you're summing over the annotations to be able to get a final estimate, which essentially means that, especially if you have a lot of annotations, that's increasing the computational time quite considerably in some cases. The Bayes RC pi approach is at each individual step of this Bayesian inference procedure, it's sort of selecting which annotation should be included for a particular variant. So it doesn't significantly increase computational time compared to the Bayes-R approach. So I guess that's a long answer to say that it's certainly not as computationally efficient as a model like GBLUP, but there's a sort of natural ranking there of Bayes-R to Bayes RC pi to Bayes RC plus.
Yeah, thank you very much for this complete answer. Yes, we have two more questions from Agnieszka. So are you aware of an approach that tries to estimate prior distribution of a trait and potentially match it with the best predictive models?
I'm not. Does that exist, or is that a suggestion?
I don't know. Agnieszka, it's close to me, so maybe she knows, but
no, it's a question. I think it would be very handy to have it.
I agree. I'm not aware of something like that, but it may be that it's off my radar, but I'm not aware of such an approach.
Thanks.
What would I wonder whether the idea would be to match the particular distribution of a trait with the annotations ahead of time, or we did look a little bit in terms of trying to understand the informatability of annotations for a given trait. We did look at trying to perform some sort of prior analyses ahead of the genomic prediction to try to guess which annotations look to be most closely linked to traits, but we didn't really have anything definitive that came of that.
Thank you very much. Yeah, Agnieszka is also asking if you have tried to use genomic annotations in the model, such as annotation for coding sequences and translated regions, etc.
We did. So pretty early on in this work, we had been using as annotations exactly that type of genome, like what we classically mean by annotations, I guess, when we're talking about genomes, or splitting splitting SNPs according to their location, intronic, exonic, et cetera, beyond just like start-stop sites, et cetera. But we fairly quickly moved on to trying to use outside annotations with the aim of going towards those Geneswitch assays. So I don't have too much feedback in terms of how using the classical genomic annotations performs for the prediction models.
Thank you. We have another question in the chat. So yes, Donna is asking, does the usefulness of the method over the GBLUP erode as most of the cattle population now have more than 1 million of genotypes? So yeah, I don't know if you have any.
Yeah, I think so. I think the added benefit of these like increasingly sophisticated models that are inserting you know, prior knowledge is probably fairly minimal in cases like for cattle where there's this huge data sets that are available for learning. I guess it's possible maybe for small breeds or yeah, for other species in which the data sizes are not nearly as large, there's some maybe greater potential for gain. But that said, you know, the general results of our group, as well as other groups that are working on fairly similar developments, I think there have been fairly disappointing results across the board in terms of the potential for including prior biological knowledge into these genomic prediction models. So I'm not aware of any approaches that have just blown other models out of the water by incorporating in some manner these biological annotations. But I do think that I think that's the case that yeah, in cattle, I would expect that the very, very large data sets that are available would not particularly benefit from these approaches.
Okay, thank you very much. And Donna said that there is no benefit neither their side, so that that makes sense currently.
Yeah, it was quite interesting because these H2020 projects, there were a bunch that are part of the Eurofang initiative. So there's sort of grouping of European projects that are working on the functional annotations of animals. And there were several groups in parallel that were working on similar questions. In fact, there were very similar worded work packages on this very topic in several parallel projects. And I think everybody's had fairly similar outcomes actually. So there was a lot of early hype about where we could go with this. And I think that has been walked back a bit lately.
Great, thank you very much. I guess we have no more questions, so I think we can close the session now. That's fine to you. So thank you very much again for two great speakers. It was very interesting.